Our next speaker is Milos Filipovic from the Leibniz Institute.
Thank you. Can you hear me? Yeah. Okay, before I start, I'd like to thank the organizers for this opportunity and for a great meeting. And I'm going to talk about something that we didn't have opportunity to hear so much, and that is how post-translational modifications are affected by aging or correlate with aging. And we'll focus on one particular residue, and that is cysteine post-translational modifications. And in my lab, we have a very simplistic and naive view on aging as a process that is invert or opposite to evolution of life, and that the processes that lead to maintenance of life are probably the one which we should explore through evolutionary processes. And life actually started thanks to hydrogen sulfide, which is what brings us to the study that we do. And hydrogen sulfide, cyanide, oops, sorry. That was a wrong button. Hydrogen sulfide and cyanide were the only two required molecules together with UV light to form all the biomolecules. And actually, it was suggested that then cysteine served as the catalyst for the first proteins to be synthesized. And life was actually flourishing in hydrogen sulfide environment for hundreds of millions of years, evolving towards complexity, despite these very harsh conditions, which is extreme radiation, abundance of radicals. And we were wondering if some of those mechanisms that maintained life during that time remained imprinted in the cells. And indeed, the cells, all cells, produce hydrogen sulfide by at least 3 enzymes, and I'll draw your attention to one because it's going to be a topic of the talk, and that is cystathionine gamma liase, CSC. The way that hydrogen sulfide works is through the post-translational modification called persulfidation, and that is addition of an extra sulfur to a cysteine, which dramatically changes the reactivity of that cysteine. And we propose that persulfidation would be an evolutionarily conserved mechanism to rescue cysteines from hyperoxidation that would inevitably happen during aging or different diseases. And the way would be that sulfide would then react and form a persulfide, which could be restore back the thiol. Or persulfide would be even better scavenger of Ross, and then form the species that could also be reduced and restore the thiols. And to do that, we developed methods to monitor persulfidation, and we could really show that it is evolutionarily conserved, it's present in all life forms, and that it directly correlates with organisms' ability to fight stress, and it correlates with the ability with the health span and the lifespan. More importantly, if you knock out the gene that is making hydrogen sulfide, that is CSC, you completely abolish the well-known effects of lifespan extension, like a calorie restriction, shown here, or inhibition of the mTOR. I can tell you also that metformin effects are also completely abolished by knocking out this gene. And this correlates well with what Vladimir Gladyshev showed, that CSC is one of few genes that is upregulated in most of the lifespan interventions that he tested. Unfortunately, as we age, we lose ability to form sulfide. As you can see here, the enzymes are disappearing, and the persulfidation is dropping down. And this is even better shown here, and these are the fibroblasts from the same individual to different time points of that person's age, and you can see a decrease in protein persulfidation and a rapid increase in hyperoxidation. But correlations is good as long as we know what they mean. So we wanted to really quantify the changes, the thiol changes that are happening with aging. And this is done by Thibault, my PhD student. And we took brains from 10-week, 10 months, 18 months old mice, wild type and CSC, and then looked at all this modification. And what we could see is that indeed the thiol oxidation is strongly increasing with aging, almost linearly from 10 weeks to 18 months. And this process is even stronger in the mice lacking CSC. As you can see here, the 10 weeks old mice are actually having higher oxidized cysteines than 18 months old wild types. According with the hypothesis, the persulfidation is dropping down, several hundreds of protein targets are dropping down, and then one can do many types of analysis, enrichment pathway analysis, clustering, but we were really interested in what is the functional effect of that. So I told to Thibault to pick up a dozen of proteins that he can find a commercial assays for, and just compare the changes that we see in the persulfidome with actual activity changes, and he came sad and said there is no change. So it's not a bad thing. Maybe the effects are at the biophysical properties of the proteins, which brings us to another very old biophysical property of proteins, and that is the liquid-liquid phase separation that also led to the evolution of life. We already heard a little bit about it, and this is the ability of proteins to switch from a single phase to a droplet phase, usually caused by crowding or interaction with RNAs. And last year's breakthrough prize in life science was given for the discovery of liquid-liquid phase separation. So we compared our data with the known proteins in so-called stress granules, and we could see that there is a substantial number of proteins that are indeed prone to phase separation. Among them, G3BP2, which is the key initiator of stress granules, showed a twofold decline in persulfidation and twofold increase in hyperoxidation. And that protein has one cysteine, and most of it is, of course, intrinsically disordered regions. And to make a long story short, what we saw is that persulfidation actually controls the stress granule, the ability of cells to resolve stress granules. And that is probably best documented here, where you could see that in the wild-type cells, after you remove the stressors, the stress granules are completely resolved. But if you do not have H2S, you do not have persulfidation, you have a hyperoxidation, the stress granules do not resolve, and they stay there forever. And I just would like to say that the stress granules are the mechanism by which the cells actually protects the RNAs from the damage that is caused by the stress. But what we found more interesting is that within the stress granules, we also saw the GAPDH. So for those of you who are familiar with GAPDH as a loading control, that is because it is one of the most abundant proteins, but it's also one of the key glycolytic enzymes, and also very structured protein. So against the theory of having intrinsically disordered regions as a prerequisite for the phase separation. And indeed, the GAPDH showed a nice phase separation, which was even more enhanced if the cysteines are hyperoxidized, and completely abolished if it was persulfidated. And you could see here that we need basically very little of GAPDH if it's hyperoxidized to form granules. And even if we form them and then expose to hydrogen sulfide to cause persulfidation, we completely melt them within minutes. Yeah? So we wanted to know what is the mechanism of which cysteine is involved, and GAPDH has 3 cysteines, 2 in the active sites, but they seem not to be important. The ones that showed the strongest correlation with aging were actually cysteine 245, and you could see that in the CSC knockout mice, even at 10 weeks, we have much higher levels. And what this told us is that then proteins would stay longer in the phase separate, and maybe they could aggregate. And indeed, the GAPDH was found more in the aggregated fraction rather than in a soluble fraction. Then we modeled it, and what we could see is that the hyperoxidation of the cysteine 245 is actually causing the flattening. The blue is the model structure. So there's a flattening of beta sheet domain, making them stickier so that they could better aggregate. And that this is actually a common feature of hyperoxidized cysteine, as demonstrated here in this example of proteins that we found to be exclusively hyperoxidized with aging. In all of them, the cysteine is in the beta sheet domains. So to see if this is a widespread phenomena, we applied a thermal proton profiling, which gives us a global meltome of the cell. And we focus on 2 particular temperatures, 47, which would be the beginning of melting, and 57, which would be the end of the melting. And you could see here that, of course, the proteins have a certain melting curves. The CSC cells do have slighter tendency to be melt faster, but if you stress them, at 47, all your proteins are completely gone, compared to the wild types where you just start to melt them. So they are more prone to aggregate. So what does this mean on a global animal level? Well, that first means that the CSC mice, they live shorter, much shorter, and also we saw widespread neurofibrillary tangles throughout the brain and all the features associated with Alzheimer's disease, making us suggest that this may be the first model for the age-induced Alzheimer's disease in mice, which normally do not form age-induced neurodegenerative diseases. Now, there are a few companies developing hydrogen sulfide donors, and the problem is that this is a gas, so it's not easy to regulate the speed of release and everything. And we were interested in finding a compound that could actually serve as an alternative substrate for CSC, so we would have a natural production of H2S by the capacity of the cell, and that this compound we could apply on humans as well. And we did find a compound that does the same. It extends life, but more importantly, it extends the health span to the level that we have 14 days old worms swimming almost as a young adult. And then we gave them to rats, old rats, and then for 3 weeks only, and they were already running 2 times faster. Their total muscle mass was increased, the lactate levels were reduced, and the NAD+ levels were dramatically increased. And they were also increased in aged worms and cells as well. And then we went back to the persulfidome data that we obtained, and we could find a target enzyme which is modulated by persulfidation to increase the NAD+, but that's something we can discuss maybe some other time. And with this, I want to finish, and I want to acknowledge the help of all these beautiful people, and I thank you for your attention.
Thank you very much. Very interesting work. We have time for some questions.
Hey.
Oh, sorry.
I think I
You
was
can
first.
start.
I'm sorry, but I apologize. Now, I just had a burning question. So the GAPDH, I mean, that's, I guess, could be involved in redox regulation locally. Your lactate and NAD, I mean, lactate is obviously regulated by redox. So do you think that the
NAD+ effect is not due to GAPDH, I can tell you. It's due to a specific enzyme that is, producing NAD+, and it is activated by persulfidation. The effect, I think, is not really so dramatic, but what I find interesting is that by treating the worms or the mice with this drug, we do not see any change in the transcriptome, any change in the proteome. We just see changes in the post-translational level, which is persulfidation. And that's already sufficient to induce all these beneficial effects.
I think most people think that the lactate pyruvate always acts at equilibrium, right? So that means that this is definitely regulated by the redox state, right?
That's also true. That's also true, yeah.
Okay.
Do you know what happens to the persulfidome in aging C. elegans?
Yeah, that's a good question. It goes down also quite a lot, actually. We have done that as well, but it really goes down. I mean, you could show a nice time-dependent correlation with persulfidation going down with age.
Okay, thank you.
So thanks, and thanks again to all short talk speakers. And